Hi, everyone. My name is Jacob. I'm a postdoc in the Anderson Lab, and I'm going to be talking to you about AI for RNA therapeutics. So, RNA therapeutics have been around for a while. So ever since we kind of got the sense of what it meant to be to have RNA antisense binding, we got RNA for antisense oligos, RNA for degradation of mRNA, mRNA delivery itself, mRNA vaccines, CRISPR-Cas9, et cetera. And so that's been going on for the last few decades. And more recently, we've started to get quite a few approvals of siRNA therapies, as well as, of course, the mRNA vaccines. And CRISPR-Cas9 gene editing is coming soon. So just a little background on what kinds of RNA you might want to deliver. So there's a few different kinds of RNA that do different things. So one is you can do antisense oligo-mediated gene regulation. So this would just be where the whole point is you deliver some kind of RNA that hybridizes to something. And so it could hybridize to something for any number of reasons. So you can have splice switching antisense oligos whose goal is to kind of block various splice sites or cryptic splice sites to change splicing of some kind of disease-associated gene. You can bind to block, for example, the binding of a native microRNA to help block the microRNA from being active in a particular gene. You can try to activate RNase H. There's all kinds of things you can do with these short antisense oligos. And then similarly, and those mostly work by just binding to some kind of mRNA, but they usually don't have some kind of catalytic activity. But then you can also use RNAi, which does have catalytic activity, where your RNA gets loaded into a RISC complex, and the RISC complex goes and degrades quite a few RNA molecules per RNA molecule that you deliver. And so these are both these are both small RNA-based therapeutics. Another kind of strategy that you can do is genome editing. And so there you usually want to deliver both a long RNA and a short RNA. So the short RNA would be the guide RNA for doing base editing or gene editing or prime editing, what have you. And then the long RNA would be, again, depending on applications, but it would be some kind of mRNA encoding some kind of gene editor, whether that be Cas13 or dead Cas9 fused to some activating complex or some kind of base editor, any number of things. So that would be an mRNA that you'd want to deliver. There's also applications for things that could work extracellular, like RNA aptamers that bind to some particular target, where you kind of evolve a stretch of RNA. You can do this with DNA as well, but you can do it with RNA. Evolve a stretch of RNA that binds specifically to a target, kind of like an antibody. And lastly, you can do delivery of some kind of therapeutic mRNA. And so that could be an mRNA vaccine, as the vast majority of you have all probably gotten. Or it could be a delivery of some kind of therapeutic mRNA, like protein replacement therapy, if somebody has say a recessive disorder where they don't make enough of some particular important metabolic protein. So there's all kinds of RNA that you want to deliver. There's short RNAs. These can usually be chemically synthesized. There's long RNAs, mRNAs specifically. They usually have to be in vitro transcribed. And the main thing to understand about these is that these are relatively complex drugs, especially compared to something like a small molecule. And so there's all kinds of dimensions along which you might want to try to optimize and improve the situation. So, and that can range all the way from target identification, where you might be interested in finding a good epitope, say, for an mRNA vaccine, or finding a good guide RNA for CRISPR gene editing, a good antigen selection for CAR T design or for a cancer vaccine. I'll be talking a little bit more about payload design and delivery vehicle design. But then also you could imagine trying to optimize the prediction of severe adverse events. So for people that get myocarditis after the COVID vaccine, which is rare but does occasionally happen, or just more generally trying to figure out, if you give a clinical trial, how can you predict which patients are going to have the best responses and the worst responses, which can help with clinical trial design. And also just with scaling up with good manufacturing practices, that can be a complex process for these complex therapies. Yeah, and so back to the two that I'm going to be talking more about it and are more and kind of the basic biotech type of research that we do a lot of in the Anderson lab and also in the Koch. There's payload design, which is optimizing the RNA that you want to deliver. So that could be one of a variety of things. So you can optimize things to be more tissue specific or whatever. So I'll talk a little bit about, for example, optimizing RNA sequence independent of kind of what the cargo protein structure is. Um, and you could also optimize, things like RNA half-life, try to make RNA more stable because RNA is typically a pretty short-acting drug, particularly mRNA delivery is particularly pretty short-acting, which can be good or bad depending on what you desire. And then also the delivery vehicle design. So if you inject free RNA, particularly naked, unchemically synthesized, unhighly chemically modified RNA into the bloodstream, it immediately gets chewed up. So you have to put it in some kind of typically nanoparticle to keep it stable from RNases and to deliver it into cells where the RNA can get translated or break up some protein, break up some mRNA or whatever it is that you want it to do. And so these are the two things I'll be talking a little bit more about. And these are things that people have applied, specifically machine learning or AI to do. And if you're wondering what is the difference between AI and machine learning, there kind of was a difference back in the day. But these days, if you want to sound cool, you call it AI, but it's more or less the same thing as machine learning, and I'll be using the terms interchangeably. I'm sure there's a non-zero number of people that will get mad at me for that, but they're pretty similar. And the reason for this is fundamentally that modern AI is based on data. It's looking at data and trying to learn things from data. So you can do this in a couple of ways. You can do it with unsupervised learning, where you basically have a lot of data points, and you might try to say, okay, what are the patterns? So cluster analysis is an example of that. You can do supervised learning, where you are trying to predict some kind of output from input. So in this case, you might have specific data points. Each data point, say, corresponds to a patient, and you might classify the patient by whether they survived or died on some treatment or whatever. And so you might be interested in trying to figure out, like, how can we predict the patient's response to some kind of therapy? Yeah, so AI generally is about your given data and how can we learn and do interesting stuff with it. So yeah, so I figured I'd talk about now some of the applications that are coming soon to a theater near you, or in fact already present. So one of the new ones, and this is really radically interesting and a really expanding field right now, is using diffusion models and other kinds of deep learning models for protein design. And so there's a couple things you might have heard of. So AlphaFold is the first of this incredibly successful set of deep learning based algorithms. So deep learning, again, these days kind of a synonym for machine learning. It's really a subset of machine learning that uses artificial neural networks in a particular kind of context, but most of the leading models use it. So I also will use deep learning pretty synonymously with the other terms. So AlphaFold is really, really good at going from predicting structure from sequence. But what the latest models do, so-called generative models, will essentially generate a sequence that can fit to a particular structure that you want. And this is really, really useful if you're trying to do some kind of design of some cargo that you might want to deliver as an RNA therapy. So to take a very recent example here, this is from a preprint from 2022. I think Regina Barzilay and Tommy Yako are on this, so it's at least part MIT work here. What they did was they were interested in designing something that would bind to the SARS-CoV-2 spike protein. And so the SARS-CoV-2 spike protein is trimeric. And so, and it also binds to this receptor called ACE2. So theoretically, if you want to find something that binds really, really tightly to these spike proteins, you might want a trimeric version of ACE2 that binds to this thing. And so you can make kind of a trimer. And the reason you want to make it trimeric is because you can take advantage of multivalency effects in binding to trimeric spike. However, if you use a flexible linker to combine just like 3 ACE2 binding sites to SARS-CoV-2, The covalent linker is a flexible covalent linker is not as efficient as you might hope it would be, because there's entropic costs to the flexible linker arranging itself so as to allow for the trivalent binding. So what you would like to do is provide some kind of rigid scaffold that places the ACE2 mimic precisely in exactly the right conformation to bind to spike protein. And so with these protein generative models, you can now just do that. So what they did was they looked at they generated a hypothetical crystal structure for 3 ACE2 mimics binding to SARS-CoV-2 spike protein. That's what's shown on the left. And then they use a stable diffusion, they use a diffusion model to just generate a protein structure that would place the ACE2 mimics from a rigid connector all together into one protein that can bind trivalently to SARS-CoV-2 spike. And so you could deliver this, you could of course deliver this as a protein, but you could deliver it as an mRNA therapeutic as well. So generally speaking, I think that the cargoes that you can deliver as mRNA, as mRNA are going to get radically expanded in terms of the usefulness by these kind of generative models. And interestingly, if you've ever used like DALL-E 2 or whatever, this is very similar technology. So like DALL-E or Midjourney is all using diffusion models. So it's sort of porting over the image generation models you may have seen over to protein design. And so another kind of example for something where you might want to use AI for payload optimization is this nice little thing that was done a few years ago, where they're interested in optimizing the 5' UTR to improve mRNA expression as much as they could. So what they did was they basically generated a bunch of random 5' UTRs. They threw them in front of EGFP and in vitro transcribed all of them and transfected some cells in vitro with the IVT mRNA cocktail. And then they do polysome profiling to see which 5' UTRs ribosomes bind to. And then you can train a convolutional neural network, that's a particular kind of deep learning model, to try to predict based on the 5' UTR what the protein expression is, what the level of translation is going to be. And so they generated a predictive model that was able to predict extremely accurately what the expression level of a protein was going to be based on the 5' UTR. And so you can use this to design better 5' UTRs to try to improve protein expression. And you can think of this as sort of a generalized, this way of doing UTR optimization. You can also, of course, do it for the coding region. You can think of that as a generalized, kind of more effective codon optimization strategy. And then you can also think about doing more exotic things, like trying to go for a tumor, tissue, or cell cycle specific expression. Or you can, and this is a bit of a separate thing, but just in terms of the payload optimization, you can think about optimizing non-coding RNA chemical modifications. I'm not going to talk too much about that, but if you have questions, I can talk about that. And then the last thing, which a little plug for my research. So one of the main delivery techniques that's used for RNA delivery, particularly mRNA delivery, is lipid nanoparticles, which have an ionizable lipid and a variety of other lipids. They're mixed to form a lipid nanoparticle. The lipid nanoparticle provides serum stability, allows for uptake into cells. It gets endocytosed, does endosomal escape, and then the RNA escapes out of the LNP into the cytoplasm, and the mRNA gets translated into some protein, and there your therapeutic thing happens. Now, the big challenge is how good can you get at these different steps? So uptake, endosomal escape, and so on. And because this is very challenging, if you don't use a really, really good ionizable lipid, you're not really going to get much expression. And so this is something I did that I haven't seen people do yet, although I'm sure companies are working busily at it. But the strategy I did was just train a deep learning model on the chemical structures of the ionizable lipids of the lipid nanoparticles. And once you have that deep learning model that predicts activity, that predicts mRNA delivery based on structure, you go and you take 1 million lipids and you say, okay, does the model think this lipid is good? Which lipid does the model think is the absolute best at mRNA delivery? And so when we take the best ones and we test them in vivo, I found that one of the AI-generated lipid nanoparticles matches or beats state-of-the-art long mRNA delivery. And lastly, one last thing that I do not know what to make of, but is interesting, um, is this collaboration between Moderna and IBM to use quantum computing uh, to presumably optimize their lipid nanoparticles. So it could be that what they're doing there is trying to use some kind of quantum mechanical properties of the lipids to look at things like absorption, to look at things like ADMET or predict solubility. But it'll be interesting to see if that turns out to be a new frontier in these computational approaches. And yeah, with that, I will be happy to open up for discussion.
Thanks so much, Jake. Do you have any questions? I feel like we're very fortunate to have like at KI some expertise in subjects. Whenever you want to optimize your LNPs and need to do it in a high throughput way, that's Jake.
Thanks so much for sharing. I'm curious how you think about taking into account the biology that you're looking for. Like, did you train your model to think about lung delivery and how difficult or easy would it be for you or someone in a similar state to think about, muscle delivery, intranasal, systemic?
Yeah, that's a great question. Basically, you can train a model to do whatever you have data for it to do. And the data that I had was mostly in vitro. And so what in vitro mRNA delivery data will tell you about kind of is like, it won't really tell you anything about which particular organs an LNP is going to be best for. It'll just tell you like this lipid is probably going to be good for something. And so it spat out what lipid do I think is going to be the best, the best kind of generally. And so then I synthesized those and I tested them for intramuscular delivery, IV delivery, lung delivery, all these things, just kind of to see what worked. Just because there really wasn't data to be able to predict that kind of organ specificity. These lipids are also quite good intramuscular. So for like vaccine applications, they're good but not great for IV delivery. I am working on ways to generate more high throughput characterization of in vivo delivery, and of course, if you could generate those really large data sets, you can then train a model to do predictions that would be more specific.
Laura?
Can you comment on where we are about data sharing? Because in the cancer field, for instance, at some point 15 years ago, we were all super excited about stuff like TCGA, all these giant databases that one would look at and they would spit out the most predictive gene for one cancer or the other. But then it turns out that unless you have people annotating data sets very well and be willing to share, then the rest of it doesn't really work.
Yeah, so that's a great question. The kiss of death, of course, is when you see like the authors are prepared to share data. Like if you ask, like of course, then like you're never going to bother, and they're never going to give it to you, even if you ask. Not that I haven't been guilty of this before, but yeah. So it's a great question. So to get the data from my deep learning model, like I took heat maps from different papers, and I just wrote code to like scrape the colors from the heat maps and interpret those. But not all papers. So I've seen papers with like thousands of mRNA delivery data points. It's like they're not available, like the data, like they don't have like an Excel spreadsheet with all the data or whatever. So more data sharing would be really, really critical. I mean, another thing is companies have huge amounts of data that we'll never get to see. You can, so what I know about that subject is very LNP specific. So data sharing for like protein structure is actually much better because everyone puts their protein structure in PDB. And so that's probably why a big reason why these like protein diffusion models and AlphaFold and so on have been so successful, because you have this huge data set of protein structures. So then you have a huge data set for training AI models on protein structure related kind of questions. Same thing for like a lot of small molecule drug applications. There's a lot more data available, but it's unevenly shared and stuff. So certainly the more people share data and the more you can get access to data, the better the better your models are going to be. And it is sometimes a frustrating barrier that the data exists and you can't get at it.
Hello, very interesting talk. I have a few questions about like logistics of this. I'm not super well aware about this, machine learning thing, and it's kind of an extension of what she asked. So, do you need to like curate your own data sets in order to like apply directly to your application, or is that like a general guideline of generating these kind of data so that many people can use it? Not that well, as at least if you want to share it out into the public. And then second, like kind of like a follow up question is that how like in intensive are these calculations? Can you do you have to collaborate with kind of like these supercomputers, or can you do it? Is there like a end user friendly kind of like application that you could just use either web based or that can be downloaded?
Yeah, so that's the good two good questions. So your second question, like how compute intensive. So I use the Luria computing cluster here, which actually doesn't have GPUs, but there are clusters. Basically, like the it's really going to depend on the application. So the really big models for the like for like AlphaFold or whatever, training those is going to take supercomputers or whatever. Like I think there's a reason Google did it first. Using them is a lot less compute intensive. So like once the model has been trained, it's more easy to use it. I don't know if you would need like a supercomputer or if it would just be slow exactly. So for my work where you had much smaller data sets and the models were much smaller, I was able to train it without GPUs, just put it on the cluster and let it run overnight. If I had a cluster, if I had GPUs, it would probably speed up like tenfold. So it really depends on what the application is. And if the data set is big and complicated, you're going to want a supercomputer, like GPU cluster probably. And then your first question, so there's a lot of data cleaning that needs to go into it. So like if you generate a data set, you need to make sure, like I put a lot of work into making sure that like when I scrape data from heat maps and stuff, I need to wrangle everything so that everything is cross comparable. Everything kind of can get input into the same bucket. So there's some substantial kind of cleaning there and a lot of like you have to like write the code to generate the chemical structures because the papers kind of don't like output smile structures for you. So that's kind of always true whenever you're doing like a machine learning application. Like at least half of the work is kind of trying to wrangle data from different sources into similar formats.